Okay, I think I'll get started with some introductory comments. Before we introduce the speaker, just a couple orders of Society of Functional Precision Medicine business. For those of you who don't know me, I'm Tony Latai. I'm the president of the Society for Functional Precision Medicine. What you see before you is not typical Danish architecture, but rather I believe it is the Tivoli Gardens in Copenhagen, Denmark. And it's my pleasure to introduce to you the information you may have heard already that the SFPM and EHA, the European Hematology Association, are co-sponsoring a meeting in precision hematology and oncology that's going to be held at the dates shown there, September 18th to the 20th in 2024, Copenhagen, Denmark. Should be a very nice time of year to be in Copenhagen. And I think we're actually the hotel at least is named like the Tivoli Hotel or something like that. So I think it's actually quite close to Copenhagen, to this location in Copenhagen, sort of famous amusement park. So anyway, I hope all of you who are interested will save this date in your calendars. More information will arrive over the coming months with regard to the specific program, the structure, and so forth. But please plan on being there. I think this should be an excellent meeting. Moreover, what we hope it will be is sort of the founding of a meeting. We have informal plans with EHA to sponsor that will alternate between Europe and the United States in alternating years. So we hope this will be the beginning of a very good relationship. As you may know, the EHA has a special working group that is very much focused on precision hematology with a very eyes-open approach to functional precision medicine. So I want to be clear, there's going to be a very high emphasis on functional precision medicine on this meeting, but we are also going to have some guests from the more conventional genomic precision medicine side of things. So that's very exciting news, and I want you to hopefully put that in your calendars. Couple other orders of business. There is an election for board positions coming up. If you wish to self-nominate or nominate someone else, this will be closing pretty soon. In fact, tomorrow. Shortly after that, we'll announce the ballot, and a ballot will be sent out only to active members of the SFPM, and then you will have a couple weeks to vote, and then we'll announce the results. The new board members will start their position on September 1st. I just want to encourage you, you have to be an active member of the SFPM to be able to vote. That means you're either an associate member, that is, you have joined as a trainee without paying an annual fee, or if you are not a trainee, we do expect you to annually pay your membership fees. If you have not done this, please go to sfpm.io and update your membership so you're a full member. We do this not only to extract fees from you, but it's also very important when we're trying to move forward as a society to be able to demonstrate as large and healthy and active a membership as possible. So I encourage you, if you are with this program of research that we are presenting in this monthly series of talks, I really hope that you will actively join us. And if there's any questions, please don't hesitate to contact us. And there's contact information on this website. Very simple address, sfpm.io. Any questions? I'll open any questions to the audience briefly here.
Tony?
Yes.
Yeah, just one question. The September meeting, September 2024, is that going to be focused only on hematology or all areas of oncology?
Although it is sponsored by co-sponsored by the EHA, it will focus both on hematology and oncology. And that's why we actually, right up in the title, you can see that we call it Precision Hematology and Oncology Meeting. So although hematology and hemonc has taken something of a lead in functional medicine because of probably the ease of getting tumors, we do embrace and will be presenting topics in solid tumor oncology at that meeting as well. Okay, so without further ado, I'll stop the share and we will move on to our featured presentation of the day. It really is my great pleasure to introduce my friend and colleague and former trainee, Joan Montero. Joan was a postdoctoral fellow in my lab for several years, and he left the lab several years ago to return to his native Barcelona, where he became a faculty member. in my laboratory, he was the initial inventor of a strategy that we call dynamic BH3 profiling, which I think he will describe to you. I won't get into it too much, but it would really open my eyes to the possibilities of what we now call functional precision medicine. And so for me, his work was really a seminal, foundation stone of a lot of work that my laboratory has progressed in since that amount of time. He initially started his primary appointment at Institute of Bioengineering, Catalonia, where he's now an associate professor equivalent at University of Barcelona. And Joan, take it away. Tell us what you're doing.
You are muted, Joan.
Yeah, can you see my screen?
Yes, I can.
Okay. So it's my absolute pleasure to be here and to be presenting our work. I'll start by thanking Tony for his nice presentation. When we think of cancer treatment, very often what comes to our mind is a patient going to a hospital to receive a certain treatment. And obviously, the idea behind giving this therapy to the patient is to eliminate the tumor. And I think that this audience would agree with me that precision medicine nowadays is basically focused on genomics. And the idea is very simple. We try to identify mutations in tumors in order to assign the best possible therapy. And one of the best examples of this approach is this clinical trial called NCI-MATCH. In this clinical trial, they analyzed around 5,000 patient samples from different types of cancer. And not surprisingly, they identified some of the most common mutations, for example, mutations in P53, KRAS, or PI3 kinase, for example. And obviously, the idea was based on these mutations to give the best treatment for the patient. But when you go to the results section of this study, at least when I read it, I was a little bit disappointed because despite doing next generation sequencing on this and all these tumors, they could only assign so only one patient out of 5 could be assigned therapy. And not only that, but most of the patients that were treated based on this classification, they already had resistant mutations that made the treatments no longer useful. So I think that we would all agree that there is plenty of room for improvement. At the end, what we're trying to do with this molecular analysis is to identify a protein that may be mutated or overexpressed in tumors in order to give the best possible treatment, with the idea of inhibiting, for example, this oncogenic protein and engaging a signaling cascade that will end up leading to cell death and killing the tumor and killing the patient. This is pretty much what we want to achieve. The problem is that this signaling cascade is not isolated, but integrated in a complex network that has different inputs. And this makes really complicated to predict a priori if a certain treatment is going to work or not to cure a cancer patient due to the complexity of the biological system itself. That's why there's a clear lack of functional predictive biomarkers, something that can accurately tell us if a certain treatment is going to work or not to cure a cancer patient. And I think that this audience doesn't need an introduction on functional assays. This has been recently reviewed by Tony. But we heard from different seminars different approaches in order to perform these functional assays. For example, using patient-derived cell lines or organoids, microfluidics, PDXs, or diaglyting on patient tumors. And nowadays, I think that functional assays are emerging as to better treat cancer. For example, there are several companies that are starting to use functional assays, and also multiple clinical studies evaluating some of these approaches. So I think that functional precision medicine is an. Today, I'm going to talk about the technique that we use in the lab, which is mitochondrial priming. This technique is based on the fact that most anti-cancer therapies used in the clinic induce apoptosis in response to DNA damaging agents, targeted therapies, and pretty much all the drugs that we use in the clinic. There's an activation of the so-called BH-friendly proteins that engage back-and-back oligomerization. Once this happens, there's a permeabilization of the mitochondrial outer membrane that induces the release of cytochrome C, SMAC, Diablo, and other proteins that will engage apoptosis formation, caspase activation, and finally lead to apoptosis. In this process, the permeabilization of the mitochondria is the key triggering event. We know that once the mitochondria is permeabilized, cancer cells are committed to apoptotic cell death. And this permeabilization is controlled by the BCL2 family of proteins. In this family, we have effectors back and back. The ones that are they oligomerize, they form pores, inducing the release of cytochrome C and other proteins that will lead to cell death. These effector proteins can be activated by BH-friendly proteins such as BIT or PUMA and inhibited by anti-apoptotics such as BCL2, BCLXL, or MCL1. There's a fourth group of proteins, the so-called sensitizers, that by themselves are not able to engage back oligomerization, but they inhibit anti-apoptotics. Therefore, they're indirectly promoting this process as well. Sorry. When I joined Tony's lab to do my postdoc, not surprisingly, the first technique that I learned was the BHC profiling. This technique was developed mostly by Jamie Ryan inside the Letai lab. And in brief, what we do with this with BHC profiling is we isolate cancer cells. This can be from a cell line, a PDX, or a patient biopsy. Then we expose these cells to digitonin. This is a detergent that forms spores in the plasma membrane and allows the internalization of the BHC peptides. Once the BHC peptides get inside of the cell, they can interact with the endogenous BCL2 family proteins, and they can induce the mitochondrial permeabilization. We can use different methods to determine the mitochondrial permeabilization that we also call MOMP. For example, we can measure the depolarization of the mitochondria or cytochrome C release, and in both cases, this will indicate cancer cells' commitment to die. One of my first observations when I started my postdoc with Tony was that when I was comparing cancer cells that were effectively treated with non-treated, I observed that in early time points, in just a few hours, cancer cells that were effectively treated became more sensitive to these BHC peptides, and particularly to the BIM peptide, that has the capacity to directly activate back and also bind to all of the anti-apoptotics. So when cancer cells were effectively treated, they became more sensitive to these peptides. So I was able to observe a shift in the dose response curve when exposing these cells to increasing concentrations of BIM. And by comparing the treated and the untreated, I was able to quantify the increase in mitochondrial permeabilization and determine a parameter that we call delta priming. And what is delta priming? Basically, it's a measurement that tells us how effective a specific treatment is to induce apoptosis. In other words, if we imagine that this ball here is a cancer cell, if we have an untreated cell or a cell treated with the wrong treatment, we're going to need a certain amount of BIM in order to induce MOMP and apoptosis. But if the treatment is effective, it will sensitize the cells towards apoptosis, so we will need less BIM in order to engage this permeabilization. The difference between treated and untreated gives us this delta priming that we use as a biomarker, a functional biomarker, to predict therapy response. In brief, what we do with NIH profiling, as an overview, we isolate cancer cells, for example, from a patient biopsy. Then we expose these cells to different treatments. It can be a single agent or a combination. Typically, we expose the cells for around 16 hours. After that, we treat with digitonin and with the BHC peptides. And then we stain for cytochrome C, or we use dyes to measure mitochondrial polarization. And we can also use cancer-specific markers. And by fluorimetry, flow cytometry microscopy, we can compare the treated and the untreated cells to determine the delta priming for every treatment. With this information, we can compare the different treatments and identify those that are going to be most effective to kill the tumor that we are analyzing. The advantage of this technique of the NIH profiling is that we can do all this in approximately 24 hours, allowing us to directly analyze patient biopsies. In fact, when I joined the lab, the technology that was available, developed by Jeremy, was a fluorescence plate reader based assay. Very briefly, what we obtained was a single cell suspension, for example, from cell lines or a patient biopsy. After that, we permeabilize, stained with JC1, and then exposed to the peptides and digitonin. After that, we obtain a kinetic trace, and we were able to obtain a BHC profiling. By comparing the different treated and untreated, I was able, using this technique, to identify the increase in priming. And in fact, I started working with cell lines. For example, I worked with solid tumor cell lines from melanoma, colon carcinoma, lung cancer, and breast cancer, exposing them to different drugs, different targeted agents that were either approved or in clinical trials. And what I observed is that after only 16-hour exposure to these drugs, I was already able to detect an increase on priming. Not to mention that at 16 hours, there's no cell death happening yet, but the changes in the BCL2 family proteins already occurred, and I was able to capture the increase on priming induced by these treatments. When I compare these predictions made with an IBH profiling with the actual cell death that we could measure in these cell lines at 72 and 96 hours, I observed that there was a good correlation between the prediction and the actual cell death occurring 2 and 3 days later. And in fact, when I performed the statistical analysis, I observed a significant correlation between delta priming and cell death. And also I performed a raw curve analysis and obtained an area of 0.96, demonstrating the excellent predictive capacity of this assay in this subset of cell lines and treatments that we tested. For those of you not familiar with raw curve analysis, it's a statistical analysis that for a given biomarker compares its sensitivity and specificity. For example, the perfect assay would be 100% sensitive and 100% specific. It would show an area of 1, and a non-predictive assay would have an area of 0.5. You can consider your assay a good predictor if the area is above 0.8. In our case, it was 0.96, so we could objectively conclude that, that this assay had an excellent predictive capacity, at least in vitro. With these encouraging results with cell lines, we decided to move to an in vivo setting. In this case, in collaboration with Karen Tchaikovsky and Ophelia Maertens, I analyzed cells from a BRAF NF1 mutant allograft. As you can see, these mice present really aggressive melanoma lesions. So what I did was to take cells from these mice and expose them to different drugs: a BRAF inhibitor, MEK inhibitor, PI3 kinase inhibitor, mTOR inhibitor, or their combinations. And what I observed is that, once again, after only 16-hour exposure of these cells to these agents, I was already able to capture an increase in priming. And for example, the best combination was the MEK inhibitor plus the rapamycin, showing a greater increase in priming. These results, these predictions made by the NIH profiling, actually correlated really well with the change in the tumor volume, so with the decrease on the tumor volume that these mouse experienced when exposed to this. So when treated with these drugs for 4 weeks. And in fact, when I compare the prediction with the change in the tumor volume, as you can see, once again, I observe a significant correlation, again, indicating that this assay had could be used to predict cytotoxicity, not only in vitro, but also in vivo. At this point, we already had good results with cell lines and in a mouse model, but the key question was if this assay could also be able to predict the clinical response. And in order to do that, as a proof of concept, what I did was to analyze patient samples, CML patient samples. As you know, chronic myelogenous leukemia is a type of leukemia characterized by the Philadelphia chromosome, the B-cell fusion protein, and is treated with imatinib, Gleevec, or more modern molecules. Back in the day, when these patients were treated, they were treated with imatinib. So what I did was to take 25 bially frozen patient biopsies and expose them to imatinib and evaluate with an IBH profiling the increase on priming. And what I observed is that those samples that show a greater increase on priming correlated with patients that achieve at least a complete hematological response, as opposed to those samples that show a lesser increase on priming that correlated with patients that did not benefit from this therapy in the clinic. When I performed the raw curve analysis, in this case, I obtained an area of 0.89, again, demonstrating the excellent predictive capacity of this assay. Finally, I also analyzed in this study some samples from ovarian adenocarcinoma patient samples, exposing them to carboplatin. And what I observed is that those samples showing a greater increase on priming correlated with patients that benefited from carboplatin in the clinic, as opposed to those that show a lesser increase in priming that correlated with patients that showing a worse progression-free survival when treated with carboplatin. We published this study back in 2015. And shortly after, Jeremy came with a new version of dynamic behavior profiling called fact-based dynamic behavior profiling that we call IBH3. The idea is exactly the same. We obtain a single cell suspension. In this case, we use molecular markers to stain the different cells. So that we permeabilize, we expose to the peptides, we fix, and we stain for cytochrome C. The advantage of this technique is because we are using a flow cytometer. In this case, we can analyze different subcellular populations, and this is really useful, especially for when analyzing primary samples. In fact, the Letai lab and other labs have extensively used this technique to find multiple treatments for different forms of cancer. In my case, back in 2016, I started a collaboration with Andy Lane at Dana-Farber. Andy was really interested in this disease called blastic plasma-saturated red cell neoplasm. It's a rare, aggressive hematological malignancy that affects the skin, the bone marrow, the lymph nodes, and the spleen. Back in the day, there was no standard therapy available, so patients had a very poor prognosis, and the median survival was less than a year in patients who presented disseminated disease. We were interested in this disease because one of the genes that is highly expressed in BPDCN is BCL2, and this pointed to the potential use of a BCL2 inhibitor to treat this what it was an incurable disease. And we were lucky because when we started this study in 2016, the first PH mimetic was approved. It was venetoclax, also called ABT-199, and was approved for CLL treatment. As most of you know, many cancer cells use anti-apoptotic proteins to block cell death. So basically, if you have an overexpression of BCL2 may bind to BIN and BAX, and this will impair the engagement of apoptosis. But if we block this anti-apoptotic protein, for example, using venetoclax, we can then release the pro-apoptotic proteins that now will be able to engage MOMB and restore apoptosis. And in fact, this drug was successfully tested with relapsed CLL patients. As you can see in this article published in the New England Journal of Medicine in January 2016, they treated several relapsed CLL patients with venetoclax, and the response was simply spectacular. Actually, the response was so good that this paper came out in January, and in April, venetoclax got its approval first by the FDA and shortly after by the EMA in Europe. So going back to the BPDCN, what I did, because of the fact that BPDCN cells show an overexpression of BCL2, was to determine with an IBH profiling if they would respond to venetoclax. I'm not going to get into too much detail. I analyzed cell line, PDXs, and also patient samples, and in all cases, I obtained a really significant increase in priming when exposed to venetoclax. I analyzed 6 patient samples, here are shown in black, and in all cases also showing a great increase in priming when exposed to venetoclax. And 2 of the samples that I analyzed corresponded to 2 patients that relapsed, that basically had very few therapeutic options. And the oncologists that were treating them, based on these results, they decided to give them venetoclax as compassionate drug use. And the clinical response was spectacular. This patient treated at the Dana-Farber, as you can see, after only 4 weeks on venetoclax, the skin lesions practically disappeared, showing a great response. And this patient treated at MD Anderson after only 25 days on venetoclax, practically the skin lesions disappear, and by PET scan, we could not detect the remains of this disease. This study got published in 2017, led into a clinical trial. I believe is still ongoing, and for me, it was a very important study because it was the first time that I could use dynamic behavior profiling prospectively to identify a new treatment for what it was an incurable disease. What we all know, the main problem when treating cancer patients is relapse. Very often, when we treat cancer patients in the clinic, we may observe a first response, but very often what happens is that some persistent cancer cells survive, and they lead to relapse. For example, in this case, melanoma patient presenting a BRAF mutation was treated with vemurafenib. In this case, it first responded, but after several months, this same patient went to the clinic with lesions that were more aggressive and no longer responsive to therapy. What we and others have found is that anti-apoptotic proteins, in fact, play a key role on the survival of these cancer cells to treatment. And this led to the development, especially after the success of venetoclax, this led to the development of multiple BH mimetics by different pharmaceutical companies. Some of these inhibitors, they encounter problems of toxicities, but I'm pretty sure that in the next year, we're going to see many of these molecules being available for clinical use, and this will help a lot to treat advanced cancer patients. One of the things I haven't told you about the NIH profiling is that so we can do basically two things with NIH profiling. One is to predict the response to treatments. That's what I showed you until now. But also, if we use some peptides that are specific for anti-apoptotic proteins that are derived from sensitizer proteins such as BAT, NOX, or HRK, we can determine changes in anti-apoptotic dependencies and guide the effective use of BH3 mimetics. So basically, we can identify if a certain tumor is using anti-apoptotic proteins in order to survive, and we can block this adaptation by using these BH3 mimetics. And in fact, I first explored this approach in collaboration with Rhys Hack, also Dana-Farber. Rhys was interested in melanoma. As you know, melanoma is a type of a very aggressive type of skin cancer characterized by different mutations. The most prevalent is the BRAF mutation that activates the MAP kinase pathway, and this led to the development and approval of different inhibitors for BRAF and MEK that are currently used to treat melanoma patients. So what I did in collaboration with Rhys Hack was to analyze a cell line presenting a BRAF mutation called A375 and expose these cells to the dabrafenib and selumetinib and determine with the NIH profiling, the changes in priming. Not surprisingly, when I treated with the BRAF or the MEK inhibitor, I observe an increase in priming with the BIM peptide. This indicates an increase in what we call overall priming. So it means that these cells, somehow they're going to undergo apoptosis. But more importantly, I perform a time course, and I observe that specifically these cells, starting at 36 hours, they start to become MCL1 dependent. I could detect this using two specific peptides for MCL1, NOXA and MS1. As you can imagine, what we did next was to combine dabrafenib with an MCL1 inhibitor. And what we observed is that dabrafenib alone was only able to kill around 20% of the cells, while the sequential combination of dabrafenib followed by the MCL1 inhibitor AZD5991 was able to multiply by 4 the effectiveness of this treatment. So basically, what happens is that when you block the MAP kinase pathway, these cells adapt using MCL1, and this leads to a limited cell death. But if you are able to block this anti-apoptotic adaptation, then you enhance the effectiveness of this treatment. We explored the mechanism underneath this MCL1 adaptation, and we found that the dabrafenib induced a decrease on the levels of NOXA that led to having more MCL1 available. As you can see, the dabrafenib treatment didn't change the overall expression of MCL1. It increased the levels of BIM and decreased the levels of NOXA. So basically what happens in a more graphical way is that in an initial state, we have these cells with NOXA bound to MCL1. When we treat with dabrafenib, the first thing that happens is a decrease on the levels of NOXA. This frees MCL1 that now is able to capture the excess of BIM produced by the inhibition of the MAP kinase pathway. And this blocks the cell death. If we add the MCL1 inhibitor, such as AZD5991 or S63845, what we do is to block MCL1, release the activators that now are free to engage back-to-back oligomerization and restore apoptotic cell death. And in fact, apart from using this strategy, we observed that this strategy was effective in vitro using this cell line and also in vivo in a xenograft model. As you can see, dabrafenib alone was only able to delay the tumor growth, while the sequential combination of dabrafenib followed by the MCL1 inhibitor was able to effectively eliminate these tumors and enhance the survival on these mice. I also analyzed metastatic melanoma patient samples in collaboration with Charles Yoon, and what I observed is that when I was exposing these samples to MAP kinase inhibitors such as BRAF, MEK, or ERK inhibitors, when there was a BRAF mutation present, I observed that there was an induction, there was an MCL1 adaptation induced by these treatments, but we could not see it when BRAF was wild type. In other words, we believe that this sequential combination could help better treat metastatic melanoma patients presenting the BRAF mutation. And hopefully, soon we will have an MCL1 inhibitor available in order to test it in the clinic. With this same approach, in my lab, we decided to explore rational combinations in pediatric cancer. For example, Clara Alcón, a talented postdoc in my lab, studied rhabdomyosarcoma, which is a pretty common pediatric solid tumor. And what she observed is that when analyzing a cell line from rhabdomyosarcoma, she observed that when she was treating these cells with vincristine, which is this standard chemotherapeutic agent used to treat this type of pediatric solid tumor, she observed that there was an MC1 adaptation, a synergy, and also a synergy with the MC1 inhibitor S63. Also, we tested this sequential combination using a PDX model. As you can see, vincristine induced also an MC1 adaptation. And when we sequentially combine vincristine followed by an MC1 inhibitor, we observed that we could actually reduce the tumor volume in these mice, indicating that probably this treatment could be useful to treat relapsed rhabdomyosarcoma patients. In parallel, Albert Manzano, a PhD student in my lab that recently graduated, studied different targeted therapies for pediatric BALL. He analyzed several potential targeted therapies that could be useful to treat especially relapsed patients. And he found that trametinib and sunitinib induce anti-apoptotic adaptations that could be pharmacologically exploited with BH mimetics such as S63 or venetoclax, especially sunitinib with venetoclax showed a greater cytotoxicity than sunitinib alone. And Albert also analyzed different PDX models, also observing these anti-apoptotic adaptations, indicating that in the future we can use these sequential combinations to better treat pediatric BALL patients, particularly when they relapse. To summarize what I told you about these sequential combinations, basically we are now using dynamic picture profiling to identify the sequential combinations of current therapies, such as conventional chemotherapy and targeted agents with BH mimetics, in order to enhance the effectiveness of these treatments. And we have different projects in this sense, and so far we have great results in different types of cancer. Going back to the dynamic picture profiling, I think one of the best recent inventions regarding dynamic picture profiling has been performed by Patrick Vola, also at the Leitai Lab. Patrick adapted the dynamic picture profiling technology to be performed in a high throughput manner using a high counter microscope. The idea is exactly the same. You plate the cells in a 384 well plate that can be read by a microscope. You expose these cells to the peptides, and then use them for cytochrome C and HOTS in order to determine the delta priming for every treatment. What you have to do in this technique is first determine the optimal concentration of beam, and then once you have the perfect concentration of beam to perform this assay, then you can run high throughput analysis to perform a drug screening on thousands of compounds. And in fact, the huge advantage of this technique is the capacity to analyze many compounds at once. In this, recently Patrick published this technology, and he showed that he was able to predict the response in cell lines, in mouse models, and also in primary samples such as colon cancer primary samples. And I think that this a great example on how to use dynamic picture profiling to find better treatments for different forms of cancer. This technique has been implemented at the Letai lab, and recently several publications coming from Tony's lab use this technique to find new treatments for different forms of cancer. And I believe that we're gonna see more improvements and more great papers coming using this technique. Just to compare the different dynamic picture profiling methodologies that I described, we have three main versions of this assay. The first version, it's a fluorescence plate reader based version. This was the first one developed by Jeremy. This has it's a low cost because a plate reader is pretty accessible. In this case, you measure MOM using the fluorescent dye JC1. You perform a kinetic trace and doesn't require a fixation. The problem of this technique, or let's say that the cons of this technique, is that you need around 300,000 cells per treatment because you're measuring a bulk fluorescence. There's no cellular selection, so you also obtain limited data, and also you need trained personnel. The second version, the flow cytometer-based dynamic picture profiling, I believe is the most widely used. It's an accessible technology in the sense that many labs have access to flow cytometry. In this case, you measure cytochrome C release, and you can use multiple molecular markers, so you can study specific subcellular populations. Also, you obtain cellular data such as side scatter, forward scatter from the flow cytometer. The problem of this presenting this technique is that it also requires hundreds of thousands of cells per treatment. It requires also an expensive equipment. Not every lab has the capacity to buy a flow cytometer, and also requires trained personnel. The third version, developed mostly by Patrick, the high content microscope one, it's a high throughput. As I said, you can use it to analyze thousands of compounds at once, which I think is great. You can, in this case, you measure cytochrome C release. It presents a low cell requirement, around 5,000 cells per drug. And you can also use multiple molecular markers, for example, to analyze different types of cells. This version, this high throughput dynamic picture profiling version, the problems that presents is that it has a high cost and specialization. You need a high content microscope that is quite expensive equipment. It also requires to determine the optimal concentration of beam before running the whole drug screening panel, and you also require trained personnel. So overall, they all have advantages and disadvantages, but one thing that they have in common is that they require centralization. This is a problem because basically you have to receive the samples in order to perform this analysis. To solve that, some years ago, I had the idea to use microfluidics with different objectives. The first one, that was before the development of the high content microscope approach. The idea was to reduce the number of cells, mostly to be able to use non-invasively obtained biopsies, such as fine needle aspirates. Microfluidics is complicated to design, so these chips are, and I can tell you by my own experience and also Alberto Manzano's experience, they're really hard to design and to optimize. But once you have the prototype design, they can be massively produced in plastic and other materials, so you can greatly reduce the cost. So once you have your design, you can get chips that are really, really cheap. Also, these chips, because you can control the inlets and especially the inlets, you can use it to automatize this assay, for example, dynamic picture profiling. And this allows the in situ analysis. For example, you can use a non-sophisticated reader in order to basically analyze your samples. So in fact, you can use this microfluidic approach to have more accessible and user-friendly assay that we can use as some sort of kit. And with this in mind, with this in mind, we developed the microfluidic dynamic picture profiling assay. And we had, for example, in the literature, we could find different microfluidic approaches precisely for precision medicine, some of which we already heard at these SFPM seminars. In our lab, what we tried to do, as I said, was to develop a microfluidic dynamic pH profiling assay. This was mostly performed by Albert Manzano, PhD student back in the day. Now he graduated, so he's now a postdoc in the lab. And we did it in collaboration with the biosensors and bioengineering group. The idea is very simple, is to obtain, to generate a chip that could generate a gradient of beam. As you can see, the design is based on 3 different layers of PDMS. With a microfluidic circuit that renders a gradient of concentrations and has 3 independent wells per column. Albert did characterize this design using a blue dye and BSA, and he found that this design produced a linear gradient that was optimal for dynamic pH profiling analysis. He also characterized the diffusion of the peptides in the wells, because obviously you want to that the peptides basically get to all of the cells inside the well, and we found that in around 90 minutes we had a full distribution of the peptide across the wells. As a proof of principle, what Albert did was to use 2 different cell lines from GIST, from gastrointestinal stromal tumor cell lines. GIST1 that presents a KIT mutation and is sensitive to imatinib, and a second cell line, GIST1-670, that presents a secondary mutation and is resistant to imatinib. And as a negative control, we use gefitinib. The first thing that Albert did was to use the flow cytometry-based dynamic pH profiling to determine the priming in these cells when exposed to imatinib and gefitinib. And not surprisingly, what he observed was that GST1 showed an increase on priming when exposed to imatinib, but not GST1-670, which is the resistant cell line. Albert also measured cell death produced by these agents, and he found that only GST1 was sensitive to imatinib, showing an increase in cell death, but no other condition was cytotoxic. With this, once he defined these two cell lines in regard of the response to imatinib and gefitinib, what Albert did next was to adapt basically to develop the readout for the assay. In this case, we use calcine to fluorescently label the cells. Basically, calcine is a dye that gets inside the cell, gets metabolized, so healthy cells present this green fluorescence. And we use TMRE to measure mitochondrial polarization. And if the TMRE is lost, it means that the mitochondria has been permeabilized and cells are starting the process of apoptosis. When he when he exposed GST1 and GST1-670 cells to increasing concentrations of BIM, what Albert observed is that when GST1 cells were treated with imatinib, they became more sensitive to the BIM peptide. And that was not the case with the GST1-670. When he quantified using the cell profiler software the percentage of positive cells, he observed that GST1 treated with imatinib show a decrease on the number of cells at lower concentration of BIM, but he didn't observe any significant change with GST1-670. Just to be clear, what we do with the cell profiler is first we use calcine to detect the alive cells, and then use the TMRE, the red channel, in order to identify those cells that are polarized. And with this information, we can perform a binary classification of apoptotic and non-apoptotic cells. It's very similar to what Patrick does with the high throughput dynamic pH profiling. When we quantify the loss of fluorescence, indicating the permeabilization of the mitochondria, what we observe is that GST1 was sensitive to imatinib, showing an increase in priming, but GST1-670 was resistant to both imatinib and gefitinib, similarly to what we observed with the FACS-based dynamic pH profiling. So this was so basically we obtained the same results, and we decided to move to the microfluidic device. We repeated these experiments. In this case, the microfluidic device only provides a linear gradient of beam, but we were able to obtain similar results with GST1 and GST1 670. When we quantified the percentage of positive TMRE cells, as you can see, we observe a shift in the curve. And when we analyze the increase in priming, once again, we observe that the GST1 cells show an increase in priming when exposed to imatinib, but not with gefitinib, and the GST1 670 were resistant to both treatments. But in general, we obtain very similar results as we obtained with the FACS-Dyna-IP2 filing. And this was the demonstration that we could use this assay to determine cytotoxicity using this microfluidic device. What we did next, in collaboration with the Sarcoma Translational Research Program and Cesar Serrano, was to analyze a primary GIST sample. Basically, it was a biopsy coming from a patient that relapsed, that basically was with imatinib and relapsed. So we decided to test different targeted therapies, apart from imatinib, ripretinib, and dactolisib. And to our surprise, this patient didn't respond to any of these agents. Ripretinib is a second-generation KIT inhibitor that inhibits KIT also when there are secondary mutations present, and dactolisib is a PI3 kinase mTOR inhibitor. But what we did using the FACS-based Dyna-IP2 filing was to use the sensitizer peptides to try to identify antipoptotic adaptations. And in fact, what we observe is that dactolisib induces a clear BCL2 dependence that we thought that we could exploit pharmacologically by combining this compound with venetoclax. And in fact, when we repeat these experiments combining the dactolisib with venetoclax, we observe a clear increase in priming with this combination, as opposed with dactolisib alone that did not show any increase in priming. As a proof of concept, what we did next was to test this same patient sample, exposing it to dactolisib and the combination of dactolisib and venetoclax using our microfluidic device. In this case, we stained for KIT with CD117, and we found that using the microfluidic device, we're able to detect the increase in priming with the combination, but not with dactolisib alone. Showing that this microfluidic device can actually identify the effective treatment for patient, in this case for this patient sample. To conclude, what we found is that this microfluidic-based Dyna-IP2 filing can identify effective treatments in cell lines, and we also tested it in a primary sample as a proof of concept. The assay requires less than 24 hours and a small number of viable cells, around 30,000 per treatment, which is pretty accessible with, for example, patient biopsies, the patient biopsies that we may obtain in the lab. This microfluidic approach we believe that it will allow the automatization and especially in situ analysis. The idea is very simple, is to be able to take biopsies from patients, if possible, non-invasively obtained, and then run this microfluidic Dyna-IP2 filing using a specific reader in order to perform in situ analysis. And we believe that with this, we can make functional precision medicine more accessible to hospitals and patients worldwide. And with this, I would finally thank you all for your attention. I would like to first thank Tony for being such a great mentor in my postdoc and afterwards. Also Jeremy, Patrick, my collaborators, Andy Lane, Rhys Hack, Javier Ramon, Cesar Serrano, Josep Samitier, and I would like also to thank this incredible team of people that I have the luxury to work with, Albert Manzano, Clara Alcon, and Fernando Martin. And with this, I'll stop sharing and I'm happy to take questions. Thank you.
Great. Thank you very much, Joan, for a really comprehensive survey of many years of work. I'll see, I do see a hand up. I'm going to use my prerogative to just ask a quick question. If I understand correctly, I think first let me say, I think you correctly assign that like in the sort of image-based technology, the dynamic pH3 profile, like high throughput an automated imager that isn't available in everybody's laboratory, like many machines. It also looked like you require some sort of an automated imager for your microfluidic device. How do those two automated imagers differ, and how is the one you're using sort of more readily available?
So at this point, we only have a first prototype. So actually, and the idea is to use, for example, like a lead detector. This could be pretty easy to obtain. But for example, compared to the high content microscope that uses, if I'm correct, uses confocal images, it's much more complex. In our case, we just need like a reader that would be. This could make it really simple.
But it's a microscope, right? It's not a well-based as it looks like you're using cell profiles, so you're identifying individual objects using a microscope, right? So this is still
at this point, right? Yeah, we're using microscopy, but in the future, I think that for example, it will be pretty. It always comes to my mind this the cell counter, different instruments that are already available in different labs where you can, for example, count different cells. So it will be the same idea, but using fluorescence, for example.
So in other words, you'd be designing a machine that was like had less like overall protocols, but it was really streamlined and designed purely for the analysis of these microfluidic devices.
Yes, basically we have an array of wells, so you can have like a detect, measure changes in fluorescence. So it would be much simpler than a high content microscope. That's the idea.
Okay, great. Let me turn to the audience. Are there any questions from the audience? I don't see any raise your hand. I saw Chris had his hand up, but perhaps I asked the question he was going to ask because I see he put it down. Are there any other questions from the audience? Okay, well, if not, let's just thank Joan for a great talk. Joan, or really forget to check your membership status and join up if you are not an active member at this time. And we look forward to seeing you at next month's SFPM seminar series.
Thanks, Juan. That was great.
Thank you all. Thank you.